Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Aleš Štrancar from BIA Separations, a Sartorius Company.
Thank you very much. Aleš Štrancar here. Just very shortly about me, working for about 30 years on the monolithic resin, chromatographic supports development, being for more than 20 years in process development of many different processes, starting more than 20 years ago with the Plasmid DNA as the first one, and many other later on. And recently, of course, a lot of work is directed towards the mRNA. I only have about 10 minutes to present. I will try to just give you basic ideas in which directions we are going with our process, and then of course everybody is more than welcome to come back to us with questions. I hope that slides are moving. So just shortly about BIA Separations. It is now part of Sartorius family since November last year. Slovenian company of Sartorius, so former BIA Separation. We are about 120 employees. We are kind of center of excellence of Sartorius for gene therapy, dealing with new column developments and also with new process developments. What else to mention? We are developing quite a number of different processes. At the moment, just downstream, but we are also going towards the upstream. So AAV was and still is one of our focuses, and Plasmid DNA and mRNA would also be, especially within last few months, our key focus. We are located in very small country, Slovenia, very near to Venice. So if somebody is going to the Venice Carnival after the COVID is treated well with the mRNA, then more than welcome to visit us. We have better wine and for sure better prosciutto than in Italy, and I hope you can at some point test all this stuff. So let's see what we have now in let's say with regards to mRNA. Producing mRNA is something which is more or less going on within last 3, 4, maybe 5 years, and of course there is still a lot to learn and how to start with the processes when it comes to industrial manufacturing. And our key efforts are always directed towards the manufacturing. We are not that much laboratory-oriented company. So when it goes to the manufacturing, some things which usually would not take care about in the lab may become very important, especially the materials and price of the materials. As well, the purity of the reagents you are using is much more important than in the lab, because impurities may go to a patient. Of course, the price of manufacturing is also very important. Possibility of cutting down certain costs in the manufacturing would also be an issue. And then contaminants of any kind, which are either coming from the raw materials or during the process, which needs to be separated away. And when separating away, we need to make sure that we don't throw away of our precious product. So there are quite some challenges in the manufacturing, and to think about platform, this would be a challenge, even I would say more tough. So why we start with the Plasmid DNA? First of all, the plasmid manufacturing at the moment is quite of a bottleneck. I know that most of the CMOs are highly overloaded, and there are also other reasons which I try to present later on. the idea of our process would be really start with the E. coli expression, do the isolation of plasmid DNA, go to the linearization, perform IVT, and then final purification. And the better you do the steps before final purification, less work with purification you would have. And now let's start with the first part, which is plasmid DNA expression. Once again, if you would like to have really everything under your control, not being too much dependent on the supply, either because of supply issues, because you need to wait, or because of the purity, the best is you start to produce the plasmid for yourself, just by yourself. We have a standard kit or a pack which can address the purification of the plasmid DNA, but when the plasmid would need to be used for mRNA, basically we suggest to insert a linearization step in between the purification of the plasmid DNA. So in this process, we show the alkylalysis where we put calcium chloride. Calcium chloride precipitate part of E. coli RNA and also helps with the filtration. We go to the weak anion exchanger. We purify the plasmid DNA good enough to be able to perform linearization. And only after linearization, we are going to take away the most problematic impurity, and these are residual proteins. The open circular and dimers are not of an issue in the case of mRNA, as would be the case maybe for transfection of the AAV, because we can linearize open circular, we can linearize also the dimers and trimers. So if you go next slide, this is basically how the preparative chromatogram looks like. We load linearized plasmid DNA on the C4 column. We get quite nice linear plasmid DNA elution peak, and the proteins would only be found in sodium hydroxide CIP. So this column really sticks any protein-related impurity very much, so we can be sure that we don't get the proteins to the next step. Why the proteins to the next step? Because they make complexes with mRNA. As soon as you make the mRNA, the protein will stick to it, highly charged positively, highly charged negatively stuff, and then this would eat away your product. And it would be not easy to purify such complex away from your mRNA product. So this is the in-process control chromatogram. The dot lines are showing the conditions or the chromatogram of the DNA which we load on the C4, which we load to the linearization step, and then after linearization step, we see basically just nice, pure, linear plasmid DNA. So this works very well, although we are not using the bovine serum albumin. We don't want to use bovine serum albumin because we would get proteins which may be problematic for the IVT step. So if we then sum up the process, so we start with the plasmid DNA, which we then purify. We can count on something between 80 to 90%, and then we do linearization. Although we were not using BSA, the linearization is still working very well. We have 100% conversion, and then we go to the C4, where we usually get more than 80% of the product. Important to say, you also need to check the purity of enzymes, especially if you would like to avoid a headache later on with the purification. So then we go to the next step, which will be the IVT and the purification of IVT mix. And when it comes to mRNA purification, there would still be some residual proteins coming from the IVT enzymes. And of course, there will be potentially some truncated, so-called truncated mRNA, double-stranded RNA, and also the IVT reagents. So we have several possibilities in place. If you have poly A tail, you can use oligo-DT column. which nicely separates most of the stuff away from whatever has a poly A tail. The other column would be again C4HLD, which very good separates the double-strand RNA away from single-strand RNA. Also some truncated stuff would be away. Prima S, very good to separate building blocks, so reagents away from the single-strand RNA. As well, it's very good analytical tool. And then styrene-divinylbenzene, which is reverse phase. If you would like to use this for the prep, be aware you need to work with higher temperatures and with organic solvents, so maybe not as good solution for the prep, but definitely very powerful analytical tool. So all these four columns can be used either for analytics or for the prep. And of course, it depends now on your IVT steps. Either you have poly A tail before or after and so on, how you combine the columns. Very shortly to touch the most expensive part of mRNA production, which is actually the IVT. The reagents like ARCA are very, very expensive, so it is good to establish a good in-process control where you can basically optimize, by which you can optimize the use of the reagent. So we are using Prima S column because it separates mRNA away from building blocks and from the reagent, and then you can also calculate about your IVT transcription. You can also calculate how many of the mRNA molecules you would get and so on. And you see, even if you lower the ARCA by half when compared to the manufacturer instructions, you can still get basically the same amount of the product with quite the same amount of the mRNA single strand, but once again, for the half of the price of the most expensive reagent. And then we go ahead. So if we have poly A tail on our plasmid, then we would basically separate the double strand, single strand with oligo-DT. Of course, we need orthogonal chromatography to basically confirm that this is the case. So in our case, we are confirming this with styrene-divinylbenzene column. So this is before oligo-DT. We see double strand RNA and also some other impurities and single strand in dilution gradient. And after oligo-DT purification, we only see basically adenosine residuals to be present and just pure single strand mRNA. Being also confirmed with dot blot. And if you look now to the conditions or to the yields, so we first perform the IVT and then purification on oligo-DT. So transcription factor should always be much over the 100. We believe that with the proper management of the IT, we can also shoot for 200 and more. The purification on oligo-DT should provide more than 90% recovery, and then the final step, the final polishing, which would, in the case of the mRNA which already inserted poly A tail into the plasmid, we would basically get between 70 to 80% recovery, and basically only need one step after the final capping. So no lengthy the precipitations, extractions, and so on. And this is, and you are done with your process. So to conclude, in very short time, we believe that it is good for very good control over the purity and also over the supply chain. It is good that you produce the plasmid by yourself. Very important is to have analytical tools in place to control and optimize all of your steps, especially the IVT step, because you can really cut down the amount or the price for your reagents. And last but not least, good orthogonal analytical control is also necessary, so you can prove that you really have a single-strand mRNA, which is also not in a complex. For this, we also check the product by NTA, so the method which works on the Brownian motion. So if the mRNA is really pure, there should be just one peak on this Brownian motion instrument. So with this, I would end, and I will try now to get to your questions.
Great. Thanks, Aleš. So we
Let's
have
start with
a lot of questions. We'll try to work through a few of these. So is the process scalable?
Yes, the process is fully scalable up to the highest possible amount you need. So I don't know, kilograms. I'm not sure what are at the moment the scales people are using, but you can definitely count on kilograms per batch.
And what titer can be expected from the process?
What titer? Usually we would dilute in the C4 at about 1 milligram per ml. Of course, if there is a need to concentrate more, you can still do concentration later on.
Okay. And then on slide 3, you mentioned poorly defined and inconsistent raw materials create issues. You say that BSA can create mRNA complexation. Is this a function of albumin or the source?
No, this is albumin. Albumin is known to make complexes. It is his duty in human body to carry in the bloodstream to carry proteins, but also some other stuff. So it sticks to mRNA as well.
What is the binding capacity with mRNA of CIMC4 HLD, and at what maximum scale have you tested this process, column size or harvest?
So basically, this, of course, very much depends. Either this is a polishing step or a purification step. So if there are no binding competitors that you only bind mRNA, or there is something else which binds with the C4, which is hydrophobic interaction, you can count between 1 to 2 milligrams per ml, also depending on the size of the mRNA.
And when taken to industrial manufacturing, what are the highest volume levels that can be accomplished for E. coli USP?
Any bioreactor which people would have at the moment. Our biggest column is 40 liter, which can basically bind, let me calculate, 40, let's say 5 milligram per ml. So multiply 5 with 40,000. So we talk about, I don't know. So our biggest column is 40 liter, and we can load 5 milligram of the plasmid per ml of this column. We can an exchanger. Of course, now also depends on the expression in the bioreactor. But this is 40, it's 200 grams, if I calculate it properly.
That's my math. Okay. What is the meaning of transcription factor with more than 100 in IVT reaction?
Yeah, in principle, if you are able to reach higher transcription factor, of course, you need less time to produce the same amount of mRNA. You need less plasmid because one plasmid would result in, let's say, if it's 100, then we would have 100 mRNA out of one plasmid DNA. So if transcription factor is transcription factor is lower, then we would need, of course, much more plasmid DNA to produce the same amount of mRNA. So the goal should be to produce as many mRNA molecules as possible out of one plasmid DNA. And the plasmid DNA is not an issue. These are the impurities which kind of kick down the transcription factor. So we checked the plasmid, it still says the same. So we are not losing plasmid. So we believe that we can go way over even 200 with the transcription factor by properly managing everything.
Okay, great. So we have a lot of really great questions. Keep asking your questions. Every question asked here will be passed to Aleš, and he can follow up with you directly. We just have time for just one more question for the live webcast, though. So the last question now is, how can you keep your SDVB at 65%? Are you using a water bath, incubator?
Yeah, on the HPLC we use basically this chamber, column chamber. If we would need to put the prep column, we would do it in the water bath in our labs. But as I said, we are not really fond to do this on the industrial scale.
Okay, well, great. Thank you so much, Aleš.
Thank you and for organizing this, and thank to everybody to take part on this presentation. As I said, it was really very short. Yeah, get back to me with any further question, and if somebody is interested for a bit longer seminar, more than welcome. Let us know. We'll try to approach your needs. All the best and have a great day.
Great, thanks. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Bye-bye.